Welcome to microbiology. We're going to start with Chapter 1, the microbial world and you in Tortora. This video is a summary of Chapter 1 in your Tortora book. And so we're going to start fittingly with a definition of microbiology. Well, micro means small, bios means life, and ology means study of something. So we're going to study small life. Basically, that's what microbiology means. We're going to take a look at microbes in our lives. Microbes, we call them microbes because we can't really see them with the unaided eye. That means we need to have a microscope to take a look at them. Germ is a term that some people use, and it usually refers to a rapidly growing cell. Microbes include a variety of different organisms, include bacteria. That's what most people think when they hear microbiology, they think, oh, bacteria. Well, there's more to that. There's also fungi, protozoa, algae, viruses, of course, and even some other forms of microbial organisms. So we're going to take a look at microbes in our lives. The vast majority of them are actually the good guys. So most of these microbes, they are rather positive. Actually, there's only a few that are pathogenic, and that means disease producing. They're really in the minority, but because they bother us and they cause disease, we tend to kind of focus on them. The vast majority of microbes are actually doing good things. For example, they can decompose organic waste, they can generate oxygen, and they can produce chemical products, a variety of them. They can ferment foods, which we usually like. Everybody likes bread and cheese. They also make beer and wine. So there's a lot of good things that microbes will do for you. And here are also lately they're used in biotechnology to produce, for example, human insulin. We'll get to that. Here are some examples of what microbes can do for you. They can produce dyes that can be used for jeans, for example, dyeing your clothing, for example. Here's some other examples of stuff that bacteria can produce. And what you should know about microbes, why it's important to know about microbes is if you know what microbes can do to your food, you can prevent food spoilage, for example. If you know about the pathogenic ones and you know how to protect yourself from them, you can prevent disease. If you know how the transmission works, you can also prevent epidemics or pandemics even. Naming and classifying microorganisms. The dual name system was established by Linnaeus, and it's basically that you have, just like we all have a first name and a last name, same thing. So you have a genus and a specific epithet. So, for example, Staphylococcus aureus, you have the genus and then the species name. So scientific names, here are some examples, and that's something, sort of a ground rule that you should be aware of. The scientific names are italicized or underlined, and that's sort of a general rule. The genus is capitalized and the specific epithet is in lowercase. So I'll give you some examples in just a sec. A lot of times these names are Latinized. They are somewhat descriptive of what something looks like, or they refer to the name that the scientist that first described them. So here's some examples. Staphylococcus aureus is always a popular one because it's so descriptive. First of all, take a look at the rules here. The name is the whole genus and species name is italicized here. The genus is also capitalized, and it's descriptive. Staphylo means clusters. Coccus means those are round cells, round bacteria. And aureus comes from the Latin word for aureum, which is gold. So this name here describes that these are bacteria that are round. They tend to come in clusters and they have a golden color to them. Here's another example where the species name refers to or honors the discoverer that first described them. So that would be Escherichia coli. Again, take a look at the application of the rules. You have the genus is capitalized. The whole name is the whole scientific name is italicized here, and the name here honors the discoverer Theodor Escherich, and then coli refers to where you find this bacterium, which is in the large intestinal colon of mammals. Okay, so here another set of rules. After the first use, you don't have to spell out the entire name again. So if you refer one time to Staphylococcus aureus or Escherichia coli and you've spelled it out fully once, the next time you're using the same species, then you can abbreviate it S. aureus or E. coli. Okay. Here's a list of the types of microorganisms that we will be discussing in this course. And let's take a look at some pictures here. You got some bacteria, and this first panel right here is sporangia. That would refer to fungi, fungal cells. Pseudopods, they're very typical of protozoa. And then here probably some algae. So and here would be a virus, HIV. So let's get started with some bacteria here. Take a look at a first look at some of the characteristics of bacteria. They are definitely prokaryotes. That means they have no nucleus, no membrane enclosed nucleus that holds the genetic material. They do have peptidoglycan cell walls, and they divide by binary fission. Those are some of the basic characteristics. And they have a variety of different types of metabolism. All of that we will get more detail later. Archaea are also prokaryotic, but they are in a whole different domain because their genetic material is so different from the eubacteria, the general bacteria. They don't even have peptidoglycan, so they lack peptidoglycan, which is all of the the bacteria, they have peptidoglycan cell walls. They live in extreme environments, and here are some of the archaea. We don't pay too much attention to archaea because none of them are pathogenic, and so therefore we tend to not pay too much attention to them. They're interesting though. Fungi, here are some of the basic characteristics of fungi that you can read through here. And sometimes when we think fungus, we think mushroom, but molds are in there too. And we'll talk a lot about molds, also some yeast, especially the ones that could be pathogenic. And then also there are some that are used for food production, as you might know, for bread and wine. So we'll get back to that in some other lecture. Protozoa here as a group, it's sort of a grab bag category of single-celled eukaryotic organisms that are animal-like in a sense that they don't photosynthesize. So they are true eukaryotes. They absorb or eat organic chemicals. Absorption is probably the better way to say it because they don't really eat, but they can engulf stuff with pseudopods. A lot of them, they're motile. They have these pseudopods or cilia or flagella for motility, and here you can see sort of an amoeba right there. Algae, that's another part of the protista category, but they photosynthesize, so they're single-celled eukaryotes, but they are photosynthetic, and so they're green, they have cellulose cell walls, and we have very few that are pathogenic. We'll get back to those. Viruses, that's a huge category, and everybody's talking about viruses these days, so we will be spending at least one big lecture on viruses. They really deserve a little bit more space, but I'll see what we can do. Very interesting. They're sort of at the interface between life and non-life. They cannot reproduce on their own, so a lot of people say, well, they're not cells and they can't reproduce on their own, so they are non-life. But well, we do know that they can make you very sick, and if you get infected with live virus, then you know what might be coming. So they are definitely, if you can kill it, then it must have been alive before. So there's this controversy about where to categorize them, because they are acellular. So that means they are not cells. They cannot reproduce on their own, but they sure can reprogram your cells to make more viruses. And in that sense, they can do a lot of harm. And of course, we will be talking quite a bit about viruses throughout this course. Then we have also multicellular animal parasites. What comes to mind here right away would be parasitic worms, and we will have a lab on that as well. And then here, the classification of microorganisms. The system developed by Carl Woese is this 3-domain system, and it's based entirely on sequencing similarities. He found that while we used to put we used to have the 5-kingdom system, and we would put all the prokaryotes into one category, but he found that the bacteria and the archaea are so different from one another that neither one of these categories is more similar to the eukaryotes than the other. So in other words, the bacteria and the archaea are so different from one another that they need to be in separate domains. And then here are all the eukaryotes. And so a picture that you probably have seen before looks something like this, where you have forms of life in sort of this tree that branches out. And here you can see there's sort of this common ancestor cell from which 3 branches arose. The eukaryotes that eventually had then their genetic material in a membrane enclosed nucleus. The archaea, which are very they live in very extreme environments, like in very high temperatures, or they are extreme halophiles. That means they live in extreme high salt concentration. So they are very different. Their metabolism is different, and very different types of little organisms. And then the regular bacteria here, the eubacteria, it's over here. So it is actually considered that the archaea are slightly more similar or more related to the eukaryotes than they are to the bacteria, which you can see there, sort of this branching off after they were sort of on the track for the eukaryotes for a little while. But anyway, we'll spend some more time on that as well. And now a little brief history about microbiology. There will be some names, and I'll point out to you which ones are important for this course. So the first time microbes, we were even aware that microbes existed, was when the first microscope was developed, and that was in 1673. So the first observations used a very basic kind of magnifying device, and Robert Hooke is one of those people that you should have heard about and should know what the important thing about him was he reported that living things are composed of little boxes or cells. Cellula, he calls them, little rooms or little boxes. And then the first observations were done by Anton van Leeuwenhoek, who actually manufactured the first very basic microscope, but he was able to see something with it. So Anton van Leeuwenhoek, that guy is credited with the first microscope. It looked probably something like this, very basic, very sort of mechanical here, but kind of interesting. He was able to see something actually with that, and so he saw some. He observed stuff that he picked between his teeth and scrapings of teeth scrapings and rainwater. So he was probably looking at some protozoa, and between the teeth we have a lot of bacteria actually, which you will find out. You can do that experiment once you got your microscope. Then here briefly, sort of as a sidetrack, the debate over spontaneous generation. So the debate over spontaneous generation goes something like this. So people started to be aware that there is this microbial life, and first reports came out that there are these little organisms. And then people were wondering, well, where are they coming from? And so a lot of people said, well, the organic material that they come out of, let's say a maggot coming out of a piece of meat that's rotting, sort of the meat generated this new form of life. So that's a spontaneous generation, that new life, especially small life, just arises spontaneously out of organic material. And the other people were saying, no, there was some fly that was laying their eggs into the meat, and then it sprouted out of these eggs. So they were pre-existing. There was pre-existing cells that just started multiplying, or pre-existing life that started multiplying. And so they went back and forth for some years. There was evidence pro and evidence con, and you can take a look at that and read through that. Different people that you don't need to know about out or need to know by name. They made their contributions to all of this, and then this Lazzaro Spallanzani guy, he boiled nutrient solution flasks and found out that, hey, if I boil this, there's no microbial growth, but if I don't boil it, then stuff starts growing. So there was something already in there. So if it was just coming out of organic material, then in the boiled flask there's still organic material in there, then something should come out. But if not, then well, something was already in there. So, and then the big breakthrough came basically with Louis Pasteur, and that's definitely a name that you need to know. Louis Pasteur, we will be talking about him. He also did pasteurization, it's attached to his name. So he put the whole thing to rest by creating this flask here that's still in some museum in France, I think. He had this flask, and he would put nutrient broth here, sort of on the bottom right there, and then he would boil it up, and he would find that if he would not bend the neck here of this flask, then eventually something must be falling in here and starts to grow in here, so the stuff would spoil on the bottom. But if he would bend the flask and made sure everything was boiled and sterile before, but the microorganisms, nothing could fall in there, and so he prevented microbials that are in the air traveling from falling in there, and so this flask stayed clear, nothing ever grew. And so this flask, I think, is still, as I said, in some museum, and that was sort of the final piece of evidence that, hey, spontaneous generation, that's a hoax. That's not true. All life comes from pre-existing life. So then the golden age of microbiology started sort of with Louis Pasteur's work, and then a bunch of new discoveries were made, fermentation, pasteurization. You can read through that on your own. And here's Louis Pasteur, what's one of his flasks. So Pasteur demonstrated that he could prevent spoilage of food by heating food up, and so we get pasteurization out of that. And take a look at your milk and your orange juice. Pasteurization is heavily used nowadays. They do ultra high pasteurization, which is that you are for a very short time, you're heating up your orange juice or whatever for very high temperatures, and that kills those spoilage bacteria, and it actually increases the shelf life of your food items by quite a bit. So I think I'm going to break this down into another section and stop here real quick.